Single-molecule protein-induced fluorescence enhancement can complement single-molecule FRET measurements when studying protein structural subpopulations and conformations, especially in cases when distinct structural subpopulations report on stable local structures.
The main advantage of this technique is that it captures distinct site-specific structural subpopulations based on the vicinity of the dye labeling site to the protein surfaces.
Single-molecule protein-induced fluorescence enhancement can be applied to any biomolecular system of interest to probe distinct local structural subpopulations.
Begin by preparing 25 picomolar sulfo-Si3 labeled α-Synuclein in measurement buffer in a low-protein binding tube. Add 100 microliters of 1 mg per milliliter BSA to the 18-chamber microscopy cover slide and incubate for 1 minute. Then, discard the BSA. Add 100 microliters of 25 picomolar sulfo-Si3 labeled α-Synuclein sample into the chamber of the cover slip slide. Next, for the data acquisition, add a drop of ultra-pure water on the top of the high numerical aperture water immersion objective lens. Fix the cover slip slide in a stage chamber, and then install the assembly on the top of the microscope stage. Bring the objective lens upwards until the water droplet on top of the objective lens smears at the bottom of the cover slip slide, and open the laser shutter. When the objective lens moves upward, inspect the pattern of the airy rings on a CCD camera. The first ring represents the focus at the water glass interface, and the second ring represents the focus at the interface between the glass and the sample solution. Increase